Welcome back to today's lesson. Mendel's observation and subsequent research promoted cell biologists to study the movement of chromosomes during the process of cell division. Plant growth and sex cell production are a result of two different types of cell division. What is that? Yes, a mitosis and meiosis. Mitosis is a type of cell division which is result in a chromosome copy, whereas a meiosis is a type of cell division which is result in a chromosome reducing. Let us see each of them one by one. The goal of mitosis is to maintain the complete number of chromosomes during cell division. Mitosis is a highly ordered process. Why? Because a chromosome loss during cell division would be detrimental to the adult plant. Mitosis can be broken into 5 basic steps, in which each are defined by the organizational state of the chromosomes. What is that? Yes, the 5 basic steps of mitosis is IP-mat, in which I is represented for interphase, P represents for prophase, M represents for metaphase, A represents for anaphase, and the last stage T represents for telophase. Let us see each of the stages one by one. Interphase is a period of cellular life when the cell grows and prepares its chromosome for cell division. It is an undividing state with 3 substages. What is that? Yes, cells grow in size and organelles are replicated during this stage, and a replication of DNA and the synthesis of protein associated with DNA. Lastly, synthesis of protein associated with mitosis also happen during the interphase stage. The second stage of mitosis is prophase. In a prophase, the chromosomes condense into the tightly wound state and the nucleus breaks down. The chromosomes appear in a disorganized mass that can be seen under the light microscopes. 3 major events happen during the prophase phase. What is that? Yes, chromosome is condensed, spindle fiber is formed, and chromosomes are captured by spindle fiber. In a third phase, which is metaphase, chromosomes become organized along the middle of the cell, and the chromosomes line up at the center of the cell with each of sister chromatids on opposite side of the cell metaphase plate. In anaphase, sister chromatids are separate. Spindle fibers attached to kinetochores shorten and pull chromatids toward the poles. Free spindle fibers lengthen and push pole of the cell apart. During this phase, the cell transiently has 4 number of chromosomes. Why? Because the centromeres between the sister chromatids are broken and producing 2 chromosomes. The last phase of mitosis is that telophase. In a telophase, the spindle fiber is disintegrated. The nuclear envelope is formed around both groups of chromosomes. Chromosomes revert to their extended states, and a cytokinesis occurs, enclosing each daughter nucleus into a separate cell. The second type of cell division is that meiosis. A meiosis is nothing but it is a type of division used to make sex cell or gamete cell. What the goal of a meiosis? Yes, a goal of meiosis is to generate haploid cells which have half the number of chromosomes as the original cell. It is a 2-step process where the original cell undergoes 2 divisions in order to make haploid cell. In the first division of meiosis, the homologous chromosomes line up together and separate from one another to generate haploid cells. But in a second meiotic division, sister chromatids of each chromosome divide in a process identical to mitosis. The two meiotic division proceed in a stepwise fashion similar to that described above for mitosis. What is that? That is a condensation of the chromosomes, alignment in the center of the cell, pulled to the opposite poles, and followed by the cell division. So, what is the difference between the two divisions of meiotic? Yes, the differences occur in how the homologous chromosomes interact with one another. In the first meiotic division, the homologous chromosomes find one another and form a structure called the tetrad. During the Prophase I, the homologous chromosomes interact with one another, which allows for the transfer of genetic materials between the homologous chromosomes in a process known as crossing over or recombination. In Metaphase I is also different in meiosis, as the homologous chromosome in the tetrad straddles the metaphase plate with each chromosome on one side. During Anaphase I, complete homologous chromosomes, each with their two sister chromatids, are pulled to the opposite poles of the cell. The centromere remains completely intact as each separate homologous chromosome is pulled to the opposite end of the cell. After the cell division, each daughter cell has only one of each homologous chromosome and therefore only half of the genetic material. The first meiotic division results in a reduction of genetic material by half, and the second meiotic division are similar to that of a meiosis. The second meiotic division is exactly like to mitosis, but with half the genetic material per cell, with the chromosome lining up at the metaphase plate, with the sister chromatids on each side of the cell. Centromeres are then broken and the sister chromatids are pulled to the opposite end of the cell. This division results in the two cells with identical genetic material, which is exactly the same process as meiosis, except with the haploid number of chromosomes. Let us compare meiosis and mitosis by a table. Mitosis is a process of asexual reproduction in which the cell divides into two producing replica with an equal number of chromosomes in each resulting diploid cell. Whereas meiosis is a type of cellular reproduction in which the number of chromosomes is reduced by half through the separation of homologous chromosomes and producing two haploid cells. Mitosis occurs in all body cells, including germ cell, whereas meiosis is only occur in a germ cell or reproductive cell. Mitosis is an equational division, whereas meiosis is a reproduction division. Mitosis is a type of asexual reproduction, whereas meiosis is a type of sexual reproduction. Mitosis is genetically producing similar daughter, whereas meiosis is genetically produce different daughters. There is no crossing over during mitosis, but mixing of chromosomes can occur during meiosis. Mitosis have one number of cell division, whereas meiosis have two number of cell division. A pair of homologous chromosomes are not occur in mitosis, whereas it is occur in meiosis. A mother cell can be either haploid or diploid in mitosis, whereas it is always diploid in meiosis. Mitosis can producing two diploid daughter cells, whereas daughter cells are haploid in meiosis. A chromosome number is the same in mitosis, but it is reduced by half in meiosis. Schismata is absent in mitosis, but it is observed during Prophase I and Anaphase I in meiosis. The spindle fiber is disappeared completely in telophase in meiosis, but it do not disappear completely in telophase I in meiosis. Nucleoli is reappeared at telophase during mitosis cell division, whereas it do not reappear at telophase I during meiosis cell division. Cytokinesis occur in telophase in meiosis cell division, whereas it is occur in telophase I and in telophase II during meiosis cell divisions. Centromeres is split during anaphase in meiosis, whereas centromeres do not separate during anaphase I, but during anaphase II in meiosis cell divisions. Prophase is simple in mitosis cell division, whereas it is complicated in meiosis cell divisions. Synapsis is not present in mitosis cell division, whereas synapsis of homologous chromosomes takes place during prophase in meiosis cell division. Thank you for watching this video.